Namaste students, I am JV Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic Nature is to comparatively complex, MHC. So in our body, something enters, some foreign particle enters, then what happens? We need a signal that a foreign particle has been entered into the body. Then what is the starting phase? And what kind of reactions, what kind of activities are going on? So we discussed about the first line of defense system, second line of defense system, and the third line of defense system, innate immunity and the acquired immunity. In the acquired immunity, we discussed that there are some components of acquired immunity. Nature histocompatibility complex is also a part of acquired immunity. It is a component of the acquired immunity like antibodies, like B cells, like T cells. So major histocompatibility is a protein complex which is located on the surface of the all nucleated cell. And what is the function of this major histocompatibility complex? And what is the importance of this major histocompatibility that we are going to discuss today? This major histocompatibility complex has important role to differentiate between the self and the non-self molecules. So as I use the line that it is located on the cell membrane of all nucleated cells. It is present in the surface of all nucleated cell. So it is present in all the nucleated cells, and they are human cell. They are human cells, and human cells know what is the self and what is non-self. If they are present on the surface, then human immune system can differentiate what is the self and the non-self. So this major histocompatibility complex has important role to differentiate between the self and the non-self. If a non-self molecule like protein, peptides, carbohydrates of the bacteria, fungus, virus comes inside the body, then this major histocompatibility complex will have important role in differentiating that this is outsider and it need to be killed. So it is present in the each cell, surface of the each nucleated cell. So it shows like a flag and shows other cells that I have a antigen. Please remove this antigen. Please kill this cell. So it has important role. So we are going to discuss about the MHC. MHC is present on the surface of all nucleated cell. It plays important role to identify the antigen between the self and non-self body, intracellular recognition, and responsible for the antigen presentation also. What does it means? Histo, major histo means tissue, and compatibility refers to the living together. If, living together means the own self means self particles are living together, but non-self are not living together. So major histocompatibility complex meaning is the tissue which are living together, the cells which are living together. An MHC molecule always recognize T lymphocytes. It has important role and it represents the antigen to the T lymphocytes. There are two types of T cells, T helper cells and the T C cell, cytotoxic cells. And similarly, there are two types of major histocompatibility complex, type 1 major histocompatibility complex and type 2 major histocompatibility complex, which have binding efficiency to the T cell and T helper cell. So, what can be defined as the major histocompatibility complex? So, major histocompatibility complex is the membrane-attached protein which work on recognition of antigen between self and non-cell body and for antigen presentation. What kind of MHC molecules should be there? So one question can be also asked that are these major histocompatibility complex present on human? Yes, they are present in the human in chromosome number 6. And what is the name of major histocompatibility complex which is present in the human? The major histocompatibility complex which is found in the human is known as the human leukocyte antigen (HLA). HLA, human leukocyte antigen. So this HLA will be identified by each cell as a self molecule. So what are the types of MHC? MHC are of four types: class 1 MHC molecule, class 2 MHC molecule, class 3 molecules, class 4 MHC molecules. Class 1 and class 2 will be discussed, but class 3 and class 4 have not any kind of sufficient knowledge about and not any kind of research on the class 3 and class 4. So in this video we are going to discuss class 1 and class 2. What is the class 1 MHC molecule? Class 1 MHC molecule are a group of major histocompatibility antigen. Okay. This is the MHC molecule. It is about a 45 kilodalton molecule, and it is a type of MHC molecule. They are present on the surface of all nucleated cells, but they are not present on the surface of the nervous tissue and platelets. What is the source of what is the use of this MHC class 1 and to which type of cell these MHC molecules show the antigen? The MHC class 1 have the compatibility to the cytotoxic cells. Class 1 will show the antigens to the class 1 will show the antigen to the cytotoxic cells. And by which receptor? It binds with the CD8 addition molecules. CD8 molecule is found in the cytotoxic cell, and it is responsible for the cell-mediated immune responses. Now, structure. Going on the structure of the MHC class 1 molecule. If we talk about the structure, and it consists of chain, alpha chain and beta chain. Two the chains are there, alpha chain and beta chain. The alpha chain is known as the alpha chain and beta chain is known as the beta 2 microglobulin. Alpha chain is non-covalently attached to the beta 2 microglobulin, non-covalently. Alpha chain contains some transmembrane glycoprotein which is encoded by A, B, C gene of the HLA, human leukocyte antigen. Alpha chain is organized by three domains such as alpha 1, alpha 2, and alpha 3. Three domains are there: alpha 1, alpha 2, and alpha 3. And beta chain has a one domain, that is beta 2 microglobulin. The alpha 1, alpha 2, and alpha 3, each domain has 90 amino acid sequence. And beta 2 microglobulin is similar to that of the alpha 3. it is similar to alpha 3, and it does not contain any kind of transmembrane protein. When an antigen is internalized and processed inside the proteasome, the peptides are formed. Peptide is further loaded on the major group on the MHC class 1 molecule from endoplasmic reticulum. Actually, human leukocyte antigen is the human antigen. It actually is the MHC molecule which is present on the human, and it is a set of surface protein present on the surface of all nucleated cells. They are responsible for the graft rejection, adaptive immunity, defense against infection. Sometimes it is expressed on the cancer cell also. So I'm going to discuss about the structure of the class 1 MHC molecule here in diagram. So this is the diagram of the class 1 MHC molecule. Um, Here there are 4 domains. This is the domain which is cytoplasmic domain. It is a domain which is known as the transmembrane domain. It is the domain which is known as the membrane proximal because it is near to the membrane, so it is known as the membrane proximal region. And this domain, which is known as the membrane distal region, because it is far away from the membrane, so it is known as the membrane distal region. In the MHC class 1 molecule, there are 4 domains: alpha 1, alpha 2, alpha 3, and beta 2 microglobulin. There is a disulfide bond between the 2 domains, and in the alpha 1, it is an glycosidic bond. This region is known as the CD8 binding site, where a cytotoxic cell will bind to this region by CD8 receptor. CD8 receptor will bind here to MSC molecule, and this is the extracellular space. This region is known as the peptide binding groove. This region is known as the peptide binding groove, and on this peptide binding groove, MSC molecule present the antigen to another cells. It present the MHC antigen to another molecule. So how the antigen will bind here? The antigen will bind here by anchoring. Anchoring means one end is here and the second one is here, and they will bind in this manner, like this here, like here. So the antigen will bind in the anchoring. The 2 ends of the antigens are binding here in the class 1 molecules. So this is the structure of the class 1 MHC molecule. So in this video, I discussed about what is MHC molecule, what are the types of MHC molecule, what is human leukocyte antigen, and class 1 MHC molecule structure and function. So students will be aware about all these topics and what kind of questions can be asked here. Students can understand the questions related to the MSC molecule. So which questions can be asked? Explain major histocompatibility complex. Explain class 1 MSC molecule. Explain structure and function of class 1 MSC molecule. Explain the complete MSC molecule including class 1 and class 2 structure and function. So these questions can be asked here. So I explained the major histocompatibility complex in this lecture. In the upcoming video lecture, I am going to discuss about major histocompatibility complex class 2 MHC. So my lecture is over here. Thank you very much.